Hola, yo soy Dulce Díaz, quizá me conoces como Pregúntale al biólogo en todas estas redes sociales y en mi canal hablamos de los seres vivos con un poco de humor, haciendo experimentos divertidos, parodias, analogías, monólogos, en fin, de todo un poco. Lo importante es divertirnos. Y te quiero contar que los talleres de ciencia han sido parte importante de mi formación en esto de hacer divulgación científica, ya que te ayudan a romper un poquito esa barrera de que solamente las personas súper eruditas mentalmente pueden hacer ciencia, cuando en realidad es que es parte de todos y está en nuestra naturaleza sentir curiosidad por las cosas y querer ir un poco más allá. Entonces, si tienes la oportunidad de participar en alguno, hazlo. No tienes nada que perder, está muy, muy padre.
Hola, buenos días, buenas tardes a todos y todas. Mi nombre es Ana Patricia Ruiz y voy a estar moderando el webinar del día de hoy. Antes de pasar a nuestra presentación, quisiera agradecerles a todas las personas que han donado y apoyado a clubes estos últimos días. Si no tuvieron la oportunidad de donar la semana pasada, no se preocupen porque todavía pueden seguir donando, todavía está el link. Nos pueden apoyar, como hemos mencionado, no importa, cada peso, cada dólar nos cuenta, así que ojalá puedan seguirnos apoyando para poder llegar a más estudiantes en México. Ok, entonces pasamos al webinar del día de hoy, que es acerca de la generación de nuevas neuronas en cerebros de personas mayores. Entonces, nuestra invitada del día de hoy es la doctora Odette Leiter. Ella estudió en Alemania, en la Universidad Técnica de Dresden, y bueno, ahí estuvo estudiando biología molecular y después se especializó en neurociencias. Actualmente, bueno, terminó su doctorado allá y después se vino aquí a Australia, a la Universidad de Queensland, en donde está estudiando, bueno, no está estudiando, perdón, en donde está haciendo investigación acerca pues de los que nos va a hablar el día de hoy, ¿no? Y cómo el ejercicio nos ayuda a todo esto. Entonces espero que, espero que se puedan motivar para después hacer mucho ejercicio y, bueno, seguir produciendo más neuronas. Hello, Odette. How are you?
Hello, good. How are you?
I'm fine. I'm fine. Thank you so much for coming today. Yeah. Well, can we see your presentation?
All right. I'll just start sharing my screen.
Great.
Do you see that?
Yes, yes.
Okay. So I'll just start, yeah?
Yeah, all yours.
Thanks for the great introduction. I heard a few bits of it. My Spanish is not too good, so hopefully all of you can understand when I present this talk in English. And I would like to talk a bit about a process that is called adult hippocampal neurogenesis, which is the formation of new neurons in our adult brains. And we would like to use this process of new neuron formation to potentially improve cognitive function during aging. So the concept of adult neurogenesis or new neuron formation in our adult brains is a fairly new one. So it hasn't been known for a long time. Only in 1963, Joseph Altman was the first one who found cells that were dividing in the brain. Do you see my pointer? Do you see the mouse?
Yes, we can see it.
Yeah. Okay, so here on the right image, there's a cell in the brain. And the cell is labeled with these black dots, and that's a thymidine analog. So thymidine is incorporated into our cells when they divide. And yeah, so during cell division, we can basically say, yes, this cell is a cell which just divided now at this given time. That means it's a cell that has been produced newly in our adult brains. But Joseph Altman had a bit of trouble to get this research, to convince people that this is actually true, because there was a dogma which basically said, or which was said by Santiago Ramón y Cajal, who was a brilliant neuroscientist at his time, and he said a sentence: once development was ended, everything may die and nothing may be regenerated. But this was only because back at the time, which was in the early 20th century, that basically he didn't have the tools to actually see these dividing cells. And the problem is that his statement wasn't cited completely, because he actually said it is for the science of the future to change this statement and to find these dividing cells. However, after Joseph Altman's discovery in 1963, it took another 30 years, so almost 3 decades, to actually get this this thought of new neuron production in the brain accepted in the scientific community. And this is mainly also because we weren't quite sure if this only happens in rodents. So the study of Joseph Altman was done in rodents. However, something happening in rodents doesn't mean that it's important for us humans. And then in 1998 as well, there was a study showing that neurogenesis or new neuron production can also occur in the human brains. And another very interesting study that I want to point out here was published only in 2013, basically showing that we have these newly formed neurons in our brains. And the basic, the study or the content of this study was nicely illustrated here by Gerd Kemperman, who basically showed that the Swedish team of Jonas Friese here that published this paper used a very elegant way to show when the cells in our brain were actually produced. So the study uses radioactive labeled carbon, which is present in our atmosphere. And this is because there was a lot of above ground nuclear weapon testing in the middle of last century. So this radioactive carbon is in our atmosphere and is taken up by plants and animals. And we then eat these products, which means that we also carry radioactively labeled carbon in our system. So this is incorporated into our cells. However, the radioactive carbon is also decaying. So we can kind of tell by the level of radioactivity, which is quite low, so it's not really harmful for us. But by the level that the radioactive carbon has in our cells, we can basically say at what time this cell has been born or produced in our brain. Yeah, and because in the process of new neuron generation, which we call neurogenesis, we just one second. Sorry. I just got instructions to hide that, but now my screen is gone, Anna.
No, you're still there, so just share the presentation again. Sorry, and that now is great. Thank you, sorry.
Okay, so basically the radioactive labeled carbon is incorporated in our brains, and we can tell exactly when these cells were born by the level of radioactivity these carbon molecules have. So this was a really elegant study in 2013. However, it only showed that cells were dividing, but we didn't really know whether these dividing cells would also become mature new neurons. And this was something that was shown, luckily for us, for our research, in the last couple of years. So 2018, 2019, we had a lot of different studies coming out showing that the cells actually also mature, become from a dividing cell to an immature neuron, and then become a mature neuron as well. So we can find new neuron generation, not only the cell division, but also the generation of neurons. And this was really exciting. So let's have a closer look at what actually happens with these progenitor cells that become new neurons. So this scheme shows it's supposed to be a mouse brain. So this is how a mouse brain would look from the side. This is an illustration of it. And in the mouse brain and also in the human brain, we have different neurogenic niches. In these niches, we have specialized environments where so-called stem cells or neural precursor cells reside. And these neural precursor cells can become activated, divide. So they usually reside in there as a dormant cell. That means they are kind of sleeping. They can become activated, start cell division, and then mature into these immature neurons, all the way into mature neurons, which then integrate in the hippocampal circuitry. And I mentioned that there's few neurogenic niches. Importantly, the two major ones are the subventricular zone, which you can see here, and the one we are interested in is the hippocampus. Why we are so interested in the hippocampus? It is a very important region for learning and memory, and this is also one of the regions that first fails when we look further into aging, develop dementia, or neurodegenerative diseases. So a small overview on what this process of new neuron generation in the hippocampus is important for. So I already said learning and memory, especially reversal or flexible learning, also spatial learning, but also some other factors, and only a few are listed here, such as also emotional pattern separation. So we're more interested in the memory and learning part of it. And for us, as an example, the most important, or how to explain what these new neurons are doing in our brains, it's basically, if we consider a situation here like the parking lot, I basically know where the parking lot is and I know how it looks, but every day I need to flexibly update my cognitive map to remember where I parked today. If you're not parking in the same spot, this can be very tricky. And that's exactly one of these functions that the new neuron generation is important for, to update our known cognitive maps. However, neurogenesis does occur throughout life. So we have neurogenesis in the hippocampus going on from very small age, or actually in the womb of the mother already. So there's a lot of neurogenesis going on. Most of us know that, but it persists throughout life, which is the good news. However, the bad news is that neurogenesis also significantly decreases with life. So the older we get, the less new neurons we have that are produced and integrated in our circuitry. And lower levels of neurogenesis are also associated with cognitive impairment and neurodegenerative diseases. We can see this nicely in our mouse model. So this image here on the left, shows a dentate gyrus, which is a part of the hippocampus. And in this hippocampus part, we have all these green cells here, and we call them immature neurons. So they are labeled with a marker called double cortin, or here DCX. And as you can see in the young mouse, this area is just packed with these new neurons. So these are new neurons that are produced, basically a snapshot of that at a given time. When we look at an aged mouse, which is here on the bottom, the same brain region, we can only find 3 cells, 1, 2, 3. So it's very little which is going on. And we can also measure that in cognitive tests that we do with the mice. So the mice can perform learning and memory tests, whereas our young mice perform really well. Our old mice often fail to do these spatial learning and memory tests. So I already told you that aging decreases the levels of neurogenesis. However, the good news is there's a lot of different factors which can influence neurogenesis and also increase the levels of new neuron production. One of those is exercise, which I will talk a bit about it. And we also found that other cells, for example, blood cells, these are platelets, are also involved in this process. But as you can see, I listed just a few of these factors, which can also all influence the levels of new neuron production. There's a lot of factors there, and especially also lifestyle factors. It is important to know when we talk about regulation of neurogenesis and new neuron production that we want to look at these factors individually, but they all have different mechanisms, how they work. And this is just illustrated here in this example of 2 factors that I picked. One is exercise. This is when we house the mice in cages with a running wheel. And exercise increases this part of the neurogenic process, proliferation. So we have the neural precursor cells, or the stem cells, coming out of their quiescence, and they start proliferating. So this is what exercise does. On the other hand, we have environmental enrichment, where the mice live in big cages with a lot of toys and a lot of friends. And that basically is really good for the survival of these cells. In other words, if I only do exercise, I have a lot of new cells which are dividing, but I don't give the cells or my brain something to do, they might just die off. They might not become new neurons. So I kind of, even though we're studying 1 or 2 or 3 aspects, it's always good to see the whole picture. And a combination of exercise and environmental enrichment actually gives us a really nice outcome of new neuron production. And this is also something which is illustrated here. This is one of the first papers which showed that exercise had an effect on new neuron production or on the proliferation, on the cell division of the neural precursor cells. And you can see here that the authors house these mice in different environments. This is, again, this enriched environment. B is just a cage with a running wheel, and C would just be a standard cage. But as you can see, if we only look at the factor proliferation, only the runners showed an increase in the number of proliferating cells in their brains. And this paper was only published in 1999. So also the effect of exercise on new neuron production is a fairly new field. We also wanted to know, because exercise can be long or short, we wanted to know how much exercise do we actually need to improve new neuron production or start this process of proliferation, and what underlies this effect of proliferation. So we found, or others showed this as well, but we found that actually only 4 days of exercise for our mice is enough to increase neurogenesis remarkably. So here on the top we can see a standard housed mouse, which is 8 weeks old. And this image is again showing the dentate gyrus region of the hippocampus. And the black dots are all dividing cells, so they're all proliferating. So if we house these mice in a cage with a running wheel for only 4 nights, we have a lot more of these proliferating cells in this region. And when we quantify this, we can see that we have a significant increase in the number of proliferating cells. So this is really nice that we see that we only need a little bit of exercise to kickstart the whole process. And our research question here at the Queensland Brain Institute now is, how does this work? How does exercise actually increase neurogenesis? One thing I haven't told you yet is that in the hippocampus area, we also have a lot of blood vessels, and the neural precursor cells actually contact these blood vessels. So one of our questions was whether blood-borne factors that are in the blood, could have an influence on the process of new neuron production after exercise. And this is something that was shown also not too long ago. These are, well, it's actually around 10 years now. But basically, the authors here did a very elegant study where they connected the blood system of an aged mouse, here in gray, and the white mouse. So they basically just connected the blood system, and they found that when the mice were sharing the same blood circulation, the young mice actually showed a reduction in new neuron production and cognitive abilities, whereas the aged mice got better in cognitive function and also neurogenesis parameters, indicating that we have aging or immune factors which harm neurogenesis and cognitive function in the young mice. And on the other hand, we also might have rejuvenating blood-borne factors which can increase and stimulate new neuron production. So if this is happening between young and old mice, and we know that exercise can also stimulate neurogenesis, we thought maybe we also have factors in the blood that change neurogenesis after exercise. So our top questions at the time were whether the blood composition does change after exercise, whether the neural precursor cells, our stem cells, respond to these blood changes after exercise, and whether we can just identify these markers in the blood and then use these markers to increase neurogenesis regardless of doing exercise or not. To first answer the question whether neural precursor cells respond to blood following exercise, we performed a quite simple proof of principle experiment, which is called a Neurosquare assay experiment. So in this experiment, we can isolate different brain regions. In this case, we isolate the dentate gyrus region of the hippocampus where our stem cells are residing. We then dissociate the tissue, and this is shown here. So this is basically a scalpel mixing up or cutting up the hippocampus or this dentate gyrus region into very small pieces. We then have this single cell suspension. And when we grow the single cell suspension with growth factors, we get these balls of cells, which is called a neurosphere. So that's basically proliferating cells that cluster together and form these neurospheres. We can then measure the size and the number of these neurospheres, and that tells us something about the proliferative capacity and how many active stem cells we have. So we wanted to see if that measures change when we add blood from our mice that were running or standard housed. And that's what we did. We know that 4 days of exercise increases proliferation of the cells in our mice. So we isolated blood from mice that were standard housed and running and added serum to our neurosphere cultures. And we found very interesting here that when we add the blood from the running mice, we found a significant increase in the number of neurospheres, indicating that the neural precursor cells in our brain actually respond to changes in the blood that occur after exercise. We then, of course, wanted to know what are these changes and what are the neural precursor cells responding to. So we again isolated blood from mice that were standard housed and running and performed a proteomic analysis. With this, we can find out which proteins are in the blood, and we found about 600 proteins that we could determine. And of those, we found about 70 proteins, which were differently regulated or had different levels between standard housed and running mice. And then we did another analysis and found that a lot of these changes that are occurring in the blood hinted towards platelets. So platelets are the smallest cells of our blood. Here we can separate the blood into plasma and cells, and platelets are the smallest ones. And yeah, we have a lot of platelets, but it's actually, in terms of fraction, quite neglectable. But it's very interesting that platelets have a lot of interesting functions. So when I first heard about platelets, the first thing that came to my mind was wound healing and blood clotting. However, recent research showed that platelets play a number of different roles in also immune responses, tissue remodeling, and cell death, which are all very interesting when we think about new neuron generation in the brain. On the other hand, when platelets get out of control, platelet dysfunction is involved in a lot of neurodegenerative diseases. And there were also papers published which showed that platelets can influence neurogenesis, however, in different regions. This is the subventricular zone and mainly after injury, and in the developing hippocampus. But nothing was known about the adult hippocampus under physiological normal conditions, for example, after exercise. At the same time, when we found that the blood changes were related to platelets, this beautiful review paper came out highlighting what platelets actually carry. So this is a platelet here, and platelets don't have a nucleus. However, they have a lot of different components, so-called granules. And in these granules, platelets carry very interesting factors. So they have dense granules with some of these factors, but in the alpha granulus, they also have a lot of interesting factors which are regulators of growth and angiogenesis. And when we looked at this list of factors here, we also thought, okay, all of these factors, or a lot of them, are also involved in hippocampal neurogenesis. So we might have an interesting candidate here to stimulate neurogenesis after running. So we performed our Neurosphere assay experiments again, where we isolated the neural precursor cells or the primary cells from the dentate gyrus. And this time, we didn't add blood, we added platelets. So we can isolate platelets very easy by centrifugation because they're very small. The small fragments with a higher centrifugation speed will be at the ground of our test tube after centrifugation, and then the top we won't have as many. So we call the upper fraction platelet poor plasma and the bottom fraction platelet rich plasma. And when we add this to our Neurosphere cultures, as you can see here on the graph in the right, the more platelets we add, the more neurospheres we're actually getting. And this was also seen in the size of the neurospheres. So these scale bars indicate 100 micrometers. So this is a neurosphere which is about 100 micrometers in size and diameter in the control cultures. And when we added the platelets, we had many more bigger neurospheres. So this one is about 250 micrometers in diameter. And that was really interesting, showing us that our neural stem cells, although they are in the brain, can respond to the platelets, which are normally in the blood. We then performed another experiment with these neurospheres. We can just differentiate our neurospheres because the neurospheres are still neural precursor cells. That means they're pretty much stem cells. So they can still divide or differentiate into mature cells. So they can become a neuron, an astrocyte, or oligodendrocyte. And we wanted to know which of these lineages, neuron, astrocyte, or oligodendrocyte, will these precursor cells become when we add our platelets. And this is the images we get, so they're nice and colorful. You can see there's a lot of astrocytes, which are here in green, and the occasional neuron in between, which is here in red. And if we quantify all these cells, we see when we add the platelets here as platelet poor and platelet rich, we don't really have a difference in astrocytes. However, when we look at the neurons, we can see that when we add platelet-rich plasma to our cultures, we have an increase in neuronal cells, indicating that platelets do not only stimulate the proliferation, the cell division of our cells, but also make them become new neurons in our brains. So these experiments have all been done in vitro in the culture. So we also wanted to next know whether platelets actually have an important role in the running increased neurogenesis. So we performed an experiment where we depleted mice of platelets. We can do this with an anti-platelet serum. So the mice were injected with this every second day for about 2 weeks. And as you can see here on the side, after these 2 weeks, the mice still have some platelets, but not as many as our control mice. So we have 4 groups here. In our control group, they just become normal serum. We have a standard housed mouse and a running mouse, or like it's basically 10 per group. And in the group of that received the platelet depletion serum, we also have our standard house and our running mice. And both of these show that the platelet depletion worked really well. I already said that the mice were also housed in cages with a running wheel, and we chose to have them running for 10 days because we know that this is a very robust time point of proliferation. So the 4 days of exercise also increases new neuron generation or the proliferation of the neural precursor cells. But actually after 10 days, we have a really robust time point. And when we take a look at the proliferating cells in these mice, we can see that our control group showed a nice increase in the number of proliferating cells, indicating that exercise has done its job. It started the process of cell division in this hippocampus area. But in the mice that were depleted of platelets, the proliferating cells were pretty much the same. So meaning that if the mice do not have as many platelets, we also don't see increased proliferation happening after exercise, indicating that they're really important for this process. We then wanted to, of course, know why are the platelets so important for exercise. So we performed some experiments and found that platelets become activated after exercise. So basically this is resting platelets that float around in our blood, and when we do exercise, they become activated. Activation can mean a lot of different things. As you can see here, it's change of shape. That's mainly something platelets do when we have a wound and they need to close that wound. They change their shape, they become really big to really close and stop the blood flow. But this is also part of activation. Another part of activation is the release of bioactive molecules from these granules. They can release exosomes, which then can also travel to different places in the body and have bioactive functions. They can upregulate receptors and release microparticles, which also contain some of these bioactive molecules or nucleic acids. What we found in the context of exercise is that resting platelets become activated and most likely release bioactive molecules such as platelet factor 4, which I will call PF4 in the remainder of this talk. And we did different experiments with platelet factor 4. We, for example, measured the blood the blood levels of this factor or in the plasma. So we found that we have more platelet factor 4 flowing around in our plasma after exercise. And in our in vitro experiments in the cell culture, we found again that we had an increased number of neurospheres and cells that became new neurons. And that was really interesting because here we didn't use a soup of platelets that we isolated from the blood. No, we only added one single protein and we still had a quite huge effect on proliferation of neural precursor cells and differentiation into neurons. We then also wanted to know whether platelet factor 4 could increase neurogenesis in the mice because all the other experiments were in the cell culture dish. So here we used mini osmotic pumps and infused platelet factor 4 for a time of 7 days into the mice, into the hippocampus especially. And it is released at a specific flow rate over these 7 days at a concentration of 10 nanogram per milliliter. And this is the same concentration that we found important in our cell culture experiments. And here in these images, you can again see these double quarton positive imager neurons. This is in our control mice. So these are young mice. They have a lot of these cells. But in our platelet factor 4 infused mice, we have a lot more of these cells forming. So a lot more new neurons that are generated in the mice when we infuse platelet factor 4. This is also seen here in the quantification of the cells. So it's a lot of cells to count, which is a bit hard on the eyes, but yeah, it gave us pretty good results. So all of this work was published, and I'll just sum up, but if you're interested in this, you can read up on this in our paper. We basically found that platelets become activated after exercise, that the platelet-rich plasma promoted proliferation and neuronal differentiation in vitro. So we had more proliferating cells and more cells that became neurons in our cultures. We also find that we deplete platelets of the blood of the mice. The mice do not show this increase in proliferation after running. And we also found platelet factor 4, one of the platelet proteins, as an important factor to stimulate neurogenesis in our cell culture experiments and also in the mice. So to sum up this first part, we basically know that a lot of factors in the blood can influence neurogenesis after exercise. And we can now add platelets to this list of factors. And especially what we found is that the platelet reaction they show to exercise might be a physiological response, which is specific to exercise and is very different from, for example, wound healing or blood clotting, what the platelets are usually involved in. Now for the last couple of minutes of this talk, I'd like to touch on some of the human studies and how we can use these mechanisms of exercise-induced new neuron formation to improve brain health. Because we can see that exercise stimulates these new neurons in the brain. And the question is, can we use this mechanism to have these new neurons increased in old age, which then in turn can help us, with healthy aging or to keep our cognitive function running. The basic question is, can we learn from the aged mice and project that into aged humans or humans which are suffering from neurodegeneration? So I just want to point out a few studies that have been shown that exercise is really important for brain health. And one of those was performed here in 2009 by our Swedish colleagues, Georg Kuhn and his team. And they basically found that when you're fit, cognitively fit, and also physically fit at a very young age, you're less likely to have a early onset dementia or mild cognitive impairment later in life. Early onset means below the age of 65. And in fact, the group found that the greatest risks of developing these phenotypes, early onset dementia or cognitive impairment, was in these individuals who had both a very low cardiovascular fitness and also a low cognitive performance, indicating when you're young and active, you can prolong the onset of these dementias which happen later in life. But that's not only something that you can do when you're young. The good news is that exercise is also working when you're old. So it's never too late to actually start exercise. And this is what this study shows. So these are participants which are 60 to 85 years old, and they perform training for only 10 weeks. So exercise for 10 weeks at this age is actually not too much if we think about that these participants haven't been that active through their whole life. But as you can see in the exercise groups here in red, in all of these tests, they performed significantly better than the control groups, which had a really low level of, or like a normal level of performing these tests. So basically, it's never too late to do exercise. And then this is another study that we find here, because we know that in aged mice, we can just measure we can just count the amount of cells. But in humans, we can't really look at the brains and count the cells. We really have to find different measures. And one of these measures that we can use is MRI scans and determine the size and the volume of the hippocampus. And when these people perform memory tests, we can then correlate their cognitive function to their size and the volume of the hippocampus where this new neuron production takes place. So this study basically shows that exercise increases the size of the hippocampus here in both hemispheres, left and right. After a prolonged time of exercise. But still, we can still change our brains by doing exercise, which is very good news. Having a look back at our mice, we see similar results. So we can also do a little MRI with the mice. But as I said before, it's much easier to just look at the cells that are produced in the brain. So as a reminder here on the top, we have these 8-week-olds, very young mice, which show a dentate gyrus in the hippocampus, which is packed with these new neurons. These mice are very young and perform really well in our learning and memory tests. Comparison to our 24-month-old mouse, we only have a few of these cells, and the mice fail these learning and memory tests. However, when we place a running wheel in the cage of these very old mice, so these guys are 24-month-old, and they basically relate to a person which is around 80 years old. So they're at very old age. But if we give them a running wheel, we can see that we have new neurons which are formed in their brains in the hippocampus region. And these new neurons can also help to increase learning and memory function. So we see that in the mice, that the old mice which were running actually perform as well as the young mice. And that's a remarkable change in these old mice that usually fail these cognitive tests. However, we need to know, or we need to think about that when we plan our experiments, that the effect of running depends on very different things. So for example, age, running duration, intensity, and genetic background in our mice. So we can see that here in these images, we have again a 24-month-old standard mouse where most of the cells, there are some bigger cells, but most of them are really small. When they run for a little bit, so this is a medium duration of running, the mice have these new neurons which are developing, and they have a bit more of a complexity. However, only after longer periods of running for our aged mice, that's around 7 weeks of running, then they have this huge complexity in the new neurons, which look like young neurons. And the proliferation or the new neuron formation also occurs in clusters, which shows us that cell division has taken place and that many cells have come out of this running effect. So in mice, it's really easy to control all these things. We can use the mice of the same age. We can all have them running the same time. In humans, it's a bit more difficult. So in order to find an optimized duration or time for exercise, we need to do a human trial. And this is actually done here at the Queensland Brain Institute at the QBI by Perry Bartlett and Dan Blackmore. So we have an exercise study going on where we have aged participants that come in, do exercise 3 times a week, so it's quite a lot. And they do this over a course of 6 months. But the nice thing is, every month, the participants are doing cognitive testing, MRI scans, and also get their blood taken. So we can basically follow these participants and see how they improve their cognitive function. We're also, this study also has different exercise intensities. So we're trying to find the right intensity, the right duration for what's needed to improve cognitive function. And with the blood markers, this is a very elegant way as well to find whether we can find, or to see whether we can find markers in the blood of these exercising people which correlate with peaks in their exercise or cognitive performance. And that's similar to what we do in the mice, where we find that exercise is related to platelet changes in the blood. Yeah, so for our research here in the lab of Tara Walker, we're interested in, of course, whether exercise can affect platelet activity in the aged humans as well. This is something we can follow up with Perry Bartlett and Dan Blackmore in their human trial. We also would like to know, of course, whether exercise or whether platelets can increase neurogenesis and rescue cognitive decline in the aged mice. So we know that aged mice, when they do exercise, increase new neuron formation, but is that also underlying the effect of platelets like we see in the young mice? And can we then use this mechanism to combat neurogenic cognitive decline in neurodegeneration or disease models? So we're also using an Alzheimer's mouse model, for example, and see whether platelet treatment can reduce plaque formation or cognitive impairment in this mouse model. And another really interesting question is, which we're working on right now, is very simple, but how do platelets actually interact with the neural precursor cells? Because platelets are still in the blood and neural precursor cells are in the brain. And the blood-brain barrier still exists to separate these two compartments. So we're working together with Tony White's team at another institute to see whether exercise can influence the blood-brain barrier, whether platelets interact directly with neural precursor cells, can they reach the neurogenic niche, or if they release factors which then travel through the blood-brain barrier and have the beneficial effects on the neural precursor cells. Yeah, and with this, I'm at the end of my talk, and I hope I could convince you all to do a bit more exercise. I showed you today that lifestyle can influence our brain health. Exercise can increase the new neuron generation in our brains, also in very old age, and that increased new neuron generation is associated with healthier brain functions and especially spatial learning and memory. With this, I'm at the end, and I'd like to thank everyone in our lab at the Queensland Brain Institute, our collaborators, the different facilities, and the published data was all from Gerd Kempermann's lab from Germany, and of course our funding partners. And now I'm happy to take any questions.
Hi Odette, thank you so much. It was a really, really interesting talk. Now I don't know if I should do more exercise and at the same time take blood from younger people.
Yeah. If you want to donate
What?
If you want to donate your blood to science,
Yeah. Oh, maybe I could. Yes, why not? Oh, well, apparently, there's some people who's going for a run tomorrow. Yeah, and I forgot to tell the audience that actually you run a lot.
If you work on exercise and brain health, I think it's you know that it's good to run.
Yeah. So there's some questions. Okay.
Should I unshare the screen and come to the?
I'm gonna share it in the screen. So the first one, it's from Jocelyn. At what age your brain drastically decreases the production of neurons, or is it a process that occurs drastically, or there's a certain age, or it's just smooth?
Yeah. So actually, it is a drastic decrease in new neuron production. And this actually happens already in quite early age. So, so the peak of neurogenesis, of course, is after birth up to puberty. And then after puberty, we're already decreasing in neuron production. But then it kind of plateaus out. So it's not like going down all the way. There are different theories why it plateaus out and what happens. It's likely that we still have, and that's something we see also with the exercise effect. So we likely still have the stem cells sitting in the brain. We just need to find ways to activate them to form these new neurons. Yeah.
Oh, okay. So I'm already decreasing my production since a long time ago. Yeah, good to know. So the other question is a little bit more technical. I think it's related with all the images with the spots that you were showing. So the question is, how can you measure neurogenesis or the proliferation in mice?
How we measure that?
Yeah.
Yeah, so proliferation and neurogenesis are two different things, if we want to speak correct science here. So proliferation is just the cell division, and we're using different markers. So these markers are basically in our cells when they divide, and we can use antibodies which find these markers, and then we have to slice the brain into thin slices, stain these thin slices with these antibodies, and then use the microscope to visualize these slices and find the cells. And proliferating cells, the markers are usually just these small dots, which I showed. Neurogenesis is a bit different, so we're using different markers for that. Neurogenesis, one indication are the immature neurons, which were all the green cells, which I showed. So this is another marker, which is just in the cells. So we're using another antibody for that, and then count all the cells in the brain. We can also use different measures of mature neurons. So we can basically mark a proliferating cell at a given time. Let's say 4 weeks ago, I gave the mice a substance which is incorporated at the cells when they divide at that time. And then 4 weeks later, the substance is still in the cell because it was incorporated in the DNA. But 4 weeks later, I might have this proliferating cell that has developed into a neuron. So I can combine different markers. I can combine the proliferation marker from 4 weeks ago with the mature neuron marker, and can then say, okay, this cell which was dividing 4 weeks ago has now become a mature neuron. So I know it basically has produced a new neuron. Yeah. So it's all microscopy based.
Oh, wow. Yeah, you make it to look very simple, but it seems that it's quite complex. Okay, we have someone here who wants to ask you a question. Hi, hola Tere.
Hola, oye, tengo una pregunta. Mencionabas que las neuronas pueden decrecer con el tiempo y se puede igual disminuir, ¿no? Pero en algún punto la neurogénesis puede contribuir a mantener estos niveles neuronales.
You want the translation?
Yes, please.
Yeah, of course. She said that you were mentioning that at some point the neurons were decreasing, and because of aging. Okay, and also tell me if I'm doing the right translation. So the question is, doing exercise can help to maintain these levels or of this neuron counts, or it's gonna decrease?
Yeah. So there's a few answers to that. I'm trying to find the easiest one. So we know that exercise, for example, as a young age, we can have much more of these new neurons forming. And it has also been shown that if we kind of prime our system, so if we give the brain or this neurogenic region something to do, we can kind of build this reserve of new neuron generation, which then stays at a higher level during aging. So this is something we can do at young age, but if we only start at an old age, we can increase new neuron production by exercise. But in mice, for example, we see that this response also decreases after a certain amount of exercise. And this is something the human study will hopefully determine if we also have this sweet spot or this peak of exercise where we have the best result. But if we do too much of it, we actually see in mice, we see this declining again, if that makes sense. At least the response of the stem cells to proliferate, to start dividing, this is something that is only occurring for a little bit and then decreases back down. Might come up again after a certain while. Yeah. So,
go. Sorry.
Well, I was gonna say, I can imagine that. So it takes the brain a lot of energy to form these new neurons, to have these responses. That's why we always have a certain balance. So we have an increase in something, but if we have too much of it, it's just too much energy. So, it kind of is put down again, but then when we're at a level, we can always get it up again when we have enough resources and energy, I would say. It's definitely known that we do exercise at a certain level. We have a certain reserve where we can draw from, which is higher than people who are not exercising or mice which are not exercising.
Muchas gracias, Robert.
I hope that answers the question. Thanks for your question.
Gracias, Tere. Muy buena pregunta. Okay, there's more questions. There's a repeat question. It's from Jackie. What type of exercises are best for memory training? And Angel Reyes is asking the same. Is the same for other types of exercise? For example, instead of a cardio type of exercise, we could expect the same results with strength-focused training or maybe hypertrophy?
Yeah, so we are mainly working with mice. So for example, just starting with the mice, we just give them a running wheel and they start running. So that's a really high cardiovascular intensity exercise. When we give the mice a running disc, so that's not the wheel, it's just a disc where they don't have to, the intensity is just not so high. This has a different effect. So the mice need much longer with these running discs to show these responses in the brain. And the responses are also lower. For humans, I think there's also differences. And this is also something the aging study will determine. So I think I didn't tell you that there's different intensities. So there are 3 groups. In the aging, we have a low, a medium, and a high intensity group. So we want to see whether differences in aerobic exercise or stretching, if this also improves new neuron generation. The most important thing is in the mice, I think the running, or in general running, is just a peak of exercise, but every little exercise counts. It just has a different amplitude of response. And also for people, I would say you need to find the exercise that you like. So if I tell someone to run 3 times a week and they absolutely hate it, I'm pretty sure that doesn't really have the same effect in someone who really like to go for runs and this sort of stuff.
Yeah. Do you think dancing is good?
Dancing has been shown to be very good because you also have a cognitive component in it. Especially choreography dancing, you kind of have the spatial component. If you just run on a treadmill or outside, it's mainly straight. It's also good to run outside and to navigate. So that's your spatial learning and exercise in one. Yeah, but dancing has been shown, especially for older people. You can't really tell them to go out and run, so you need to find something that they can do. And a lot of people who do choreography dancing or line dance or something where you have these, you have to remember a routine as well. That has been shown to be really good for cognitive health. Yeah.
Oh, great. So it's just also different kinds of sport can play different roles in the way we train our brain.
So
Yeah.
I would say in general, moving is a good thing.
Mm-hmm.
But then, of course, there's other things like if you, play too much soccer or something and you get the ball on your head, it might
Or
be
boxing and
a negative effect. So, yeah. But on the other hand, you have a lot of running and moving. So, yeah.
yeah. There's this question from Marcel Sire. Okay, he says very cool. And yeah, this is a really interesting question. Does the adult neurogenesis occur also in regions outside the hippocampus? Because you were mentioning the hippocampus, right?
Yeah, so we were on the hippocampus because this is an important region for learning and memory, and especially in humans. That's what we try to achieve during healthy aging, to keep our learning and memory healthy. But neurogenesis has also been shown to occur in other regions. It's only a very few. Actually, in fact, there's only two which I would say have a lot of evidence. The second one is the subventricular zone. And in that region, the new neurons are used to produce olfactory neurons. So this is something that's occurring at an abundant rate in mice. So the mice have a lot of these olfactory neurogenesis because they use it a lot. They just use their sense of smell much more than us humans. In humans, I would say the rate of neurogenesis in the subventricular zone is lower compared to hippocampal neurogenesis.
What is that zone? Because the hippocampus is kind of here, right?
The hippocampus is basically on both hemispheres in our head, so it's very deep inside the brain. The subventricular zone is more in the middle, and then the olfactory center in the mice is, for example, in the very front, like close to the nose. So yeah, the olfactory neurons or neuron generation in this neurogenic niche, the subventricular zone, these neurons can also migrate all the way to the olfactory bulb to then fulfill their olfactory function of smell. Yeah.
Okay. At the beginning of your talk, you were mentioning something about mice doing exercise, but also having like a very enriched environment. So this question, I think it's related to that. Can exercise and the other factors mentioned contribute to slowing down the progression of neurodegenerative disease such as Alzheimer's?
Yeah, I think that's something that we show, because the thing is that the neurogenesis in the hippocampus or the hippocampus area is one of the regions that is first affected by these neurodegenerative diseases or complications. When we develop dementia, one of the first things that decreases is the ability of spatial learning. And we know that spatial learning is also involved with this new neuron production. So the new neurons that are produced in the area aim the updating of these cognitive maps and spatial learning. So so if we do exercise and keep our brain healthy, to train our brain, so not only physical exercise, but also cognitive exercise, environmental enrichment, so social contacts, things that lift our mood as well. So mood is also something that affects new neuron production in the hippocampus. So if we keep a general health, this of course may more prolong the onset of these diseases. And in the mouse models we could show that exercise can slow down the progression of these different diseases.
Good. And going back to the platelets, is there any therapies right now for all people so they can have like, I don't know, a transfusion of platelets and then they get better with their memory or something like that, or not yet?
Yeah, that's a good question. So I think the platelets and brain axis is a very, very new field. So we don't really know much about it yet. platelets have definitely been used to treat skin burns, so in different other regenerative contexts. So if we
Also the skin, right?
skin rejuvenation, skin burns, arthritis, because platelets carry a lot of these regenerating factors. So they have a lot of beneficial factors in them. In terms of cognitive abilities, this is a very new field. So in mice, it has been shown that platelets or the products which are produced by platelets can slow down plaque formation or Alzheimer's progression or something, for example. I'm not too sure where we are with human trials.
Yeah, that's okay. If you need someone, you can ask me, and I'll be happy to be an experiment and see if I can get smarter like that. Yeah. Well, Odette, we are running out of time. Thank you so much for giving us this talk. It's it's motivating me today. I'm definitely doing exercise, and also in the subsequent days. Okay, there's platelets are love. Okay, there's comments.
Yeah, thanks for giving me the opportunity to share my work today with you guys.
No, it's great. Yeah, and if people have more questions, could they contact you through your email or?
Sure, I'm always happy to answer questions. Yeah.
Okay, great. Well, thank you so much, and have a nice day.
Yes, thank you.
Bueno, pues muchísimas gracias a todos los que se conectaron hoy. Antes de terminar, todavía tengo más anuncios que dar. Recuerden que el próximo miércoles es ya nuestro último webinar. Y, perdón, este no es. Y va a ser Antonio Lascano, el doctor Antonio Lascano, que nos va a hablar sobre el origen de la vida. Así que, bueno, pues ojalá se puedan conectar. Va a estar muy interesante. Espero que muchas personas ya sepan de la trayectoria de Antonio Lascano. Bueno, y les recuerdo que todavía seguimos teniendo donaciones, aceptando donaciones, y esas van a ser hasta mayo. Así que esta es nuestra página. Bueno, ahorita les va a aparecer, ahí está el link. Igual, por favor, síganos en nuestras redes sociales. Tenemos Instagram, LinkedIn, Twitter, Facebook, y no sé si me está pasando alguna más, pero bueno, ahí vamos a estar anunciando otros eventos que también estamos teniendo, como son los seminarios que vamos a tener y también el podcast, que los pueden escuchar por Spotify. Entonces, bueno, pues con esto me despido y pues ahí quedamos al pendiente. Hasta luego, gracias.
Juntos somos más fuertes. Por eso en Clubes de Ciencia te invitamos a participar donando de tu tiempo y recursos para que se pueda hacer aún más impactante esta iniciativa. Yo, Claudia Larcón, embajadora de esta noble causa, te invito a hacerlo.
clubesdeciencia.mx